Hello, and thank you for tuning in. My name is Cody Smith, and I'm a third-year postdoc in the Joint Graduate Program in Toxicology at Rutgers University, advised by Drs. Deborah Laskin and Andrew Gao. Today I will be sharing with you some preliminary results of my F32-funded research investigating inflammatory signaling mechanisms in lung macrophages responding to ozone-induced lung injury. You've all probably heard about ozone in the ozone layer. Ozone in the stratosphere is great because it protects us from UV radiation, but ground-level ozone, which is generated from the reaction of automobile and factory emissions and sunlight, is a ubiquitous urban air pollutant that causes airway disease in both healthy and susceptible populations. On the right, you will see a screen grab from the EPA's website showing all of the counties in our area which had ozone levels exceeding the EPA standard of 70 ppb, underscoring the importance of evaluating the biological effects of such ubiquitous exposure. The goal of our research is to investigate signaling mechanisms contributing to ozone-induced lung injury in order to develop strategies to mitigate toxicity and to better inform regulatory guidelines for exposure. Extensive evidence from our research team has indicated a critical role for a particular type of innate immune cell, the macrophage, in regulating the acute inflammatory and later resolution phases of ozone-induced lung injury. If you're not familiar with macrophages, you can learn a lot about them from just their name, which literally means big eater in Greek. These cells are important for their phagocytic functions in clearing infections and other debris, but they also have very important regulatory roles. Tissue-resident macrophages, which spend their whole lives in the lung and are derived from the embryonic yolk sac, have important roles including regulating surfactant homeostasis and serving as sentinel cells in initiating the inflammatory response to tissue injury, resulting in the recruitment of other immune cells to the lung. Among these recruited cells are inflammatory macrophages, which are derived from the bone marrow and are characterized by the expression of CD11b. When they reach the lung, the tissue microenvironment and cytokines and mediators they encounter polarize them to either a M1 pro-inflammatory or M2 anti-inflammatory phenotype, as shown in the figure on the right. We typically consider ozone-induced lung injury to result from an imbalance of M1 and M2 macrophages, where overactivation of M1 pro-inflammatory macrophages overwhelms the suppressive and pro-resolution effects of M2 anti-inflammatory macrophages. This leads to cytotoxicity and increased lung injury in response to ozone. To identify cellular signaling pathways in macrophages that are dysregulated in response to ozone that may represent important mechanisms of ozone-induced lung injury, we performed an RNA-seq analysis of alveolar macrophages collected 24 and 72 hours after exposure to ozone and looked for enrichment of signaling pathways using Ingenuity pathway analysis software. We found that PPAR signaling was one of the most significantly enriched pathways among the differentially expressed genes in the alveolar macrophages 72 hours after exposure. Upon further investigation in the top left panel A, we found that PPAR gamma was the most abundant isoform and that its mRNA expression was significantly reduced in response to ozone. To the right in panel B, we confirmed downregulation of PPAR gamma protein expression in alveolar macrophages by semi-quantitative scoring of immunohistochemical stained lung tissue sections. In the bottom panel, we used IPA to investigate the expression of predicted downstream transcriptional targets of PPAR gamma and found that they were predominantly downregulated as indicated by the green color relative to upregulated as indicated by the red color. Altogether, these data suggest that PPAR gamma signaling is reduced in response to ozone exposure, and this is significant because extensive evidence indicates that PPAR gamma is important in promoting anti-inflammatory and pro-resolution phenotype of macrophages. This was a novel way of thinking for us, as we typically suspect that ozone-induced lung injury is a result of overactivation of the M1 pro-inflammatory macrophages, but these data suggest that it may be due to impaired activity of the M2 anti-inflammatory and pro-resolving macrophages. Thus, our hypothesis going forward is that ozone causes dysregulation of PPAR gamma signaling in macrophages, resulting in defective development of an anti-inflammatory phenotype. This leads to prolonged injury, oxidative stress, and inflammation. To investigate this, we performed an experiment to determine if administration of a PPAR gamma agonist would reduce ozone-induced lung injury. For these studies, we administered rosiglitazone by IP injection beginning 24 hours prior to ozone exposure and repeating daily. Then we euthanized the animals 24, 48, and 72 hours later and examined effects on markers of lung injury, macrophage phenotype, and inflammatory gene expression in alveolar macrophages. In the top panel, you will see the effects of rosiglitazone on ozone-induced regulation of lung injury markers, including total protein and phospholipid content. Exposure to ozone resulted in a significant increase in both total protein and phospholipid content as expected, and we found that administration of rosiglitazone significantly reduced the ozone-induced increases in phospholipid content 72 hours after exposure. In the bottom panel, we investigated effects on inflammatory macrophage phenotype. We observed significant increases in both pro and anti-inflammatory macrophages in the lung after exposure to ozone, consistent with previous studies, and numbers of both of these subpopulations were moderately reduced at 72 hours post-exposure in animals administered rosiglitazone. At the right, we examined effects on inflammatory gene expression in the alveolar macrophages. As expected, we found significant increases in the expression of anti-inflammatory marker arginase 1 and the pro-inflammatory marker PTGS2, which is also known as COX2. Consistent with the effects on lung injury markers and macrophage phenotype, we found that administration of rosiglitazone blunted the ozone-induced increases of both the anti and pro-inflammatory gene expression 72 hours post-exposure. In summary, we found that PPAR gamma signaling is altered in alveolar macrophages after ozone exposure, and that administration of rosiglitazone, a PPAR gamma agonist, blunted ozone-induced increases in markers of lung injury and expression of anti and pro-inflammatory genes 72 hours post-exposure. This collectively suggests that rosiglitazone is promoting the return to baseline and that targeting PPAR gamma may effectively dampen inflammation and tissue injury following ozone exposure. I want to thank my advisors, Dr. Deborah Laskin and Andrew Gao, and all past and present members of the Laskin and Gao laboratories who have contributed to this project. And of course, I want to thank our funding support from the National Institute of Environmental Health Sciences that made all of this research possible. Thank you.